1, 2, 3. Good. Can you guys hear me out there? Okay, so we're going to get started now. See, a few people have signed in. Today we are going to go over Lab 7 Lab. If you guys have any questions, please feel free to use the Q&A system, part of Google Hangouts. I want to thank you guys for signing on at this time. If you have any other questions and you can't use the Q&A system, please feel free to email me. I have my phone right next to me, and I will be able to answer any questions as we go along. Okay, so let's get started. So in today's lab, I know you had the other TA come in and explain to you basically what a plasmid is and whatnot. I'm just going to go over it because I know I have certain things that I want to make sure you guys really understand well without any problems, especially when it comes to taking the quiz. So, first thing that we're going to go over today is what is a plasmid. Basically, a plasmid is a piece of bacterial DNA that holds all the genetic information. Now, in this DNA, it has everything a bacteria needs in order to live. We can exploit this, and we can force the bacteria to make genes and make proteins of other genes that it doesn't normally have. And you saw that in the previous week. ADP1 had the gene for creating the enzyme to break down para-hydroxybenzoate, and we forced the ADP6 to take it up via natural transformation in order to then express that enzyme so it can live on a para-hydroxybenzoate plate. Okay. So basic anatomy of a plasma, they have these plasma maps online, and it has for every known bacteria that they know out there. And different parts indicate different things. It could be an indication of where transcription begins, where it ends, open reading frames, antibiotic resistances, enzyme production, just like the enzyme to break down para-hydroxybenzoate, and so forth. There's different types of visual indications and whatnot. This is what we would normally see online. This is the basic outline of a plasmid. And as you can see over here, this ERA-C is a gene. This PBAD, that's a promoter. GFP, the green fluorescent protein. BLA, which is beta-lactamase gene. And this ORI, origin of replication. So all I really want you to take away from this is when you see a plasmid like this, you can be able to read it and know what's going on. That this is one gene, this is another gene, this is where transcription will begin, and this is another gene over here. Okay. In Lab 7, you guys looked at green fluorescent protein. That would be this gene right over here. Okay. Now, this is something that you would see more specific online. This gives you all the information you need in order to see everything on the gene, on the plasmid. This PJEX is the name of it. PJEX, P for plasmid, 5X2. That's just a designation for this particular gene. And it has 4.9 kilobase pairs. And what does a kilobase pair mean? That means it's got 4,900 base pairs. And the base pairs is an amino acid pairing. Adenine goes with thymine, guanine goes with cytosine, and so forth. And so this has 4.9 kilo base pairs, or 4,900 base pairs. Certain anatomy of this gene, we have the SLAC gene, origin of replication. Here we have what is called a multiple cloning site, an MCS. And here this tells us that we can insert different genes into this area. And when we want to insert certain genes into it, because of different types of manipulation. Okay, and that's basically what you guys saw in Lab 7. See, we got a few new people signed on. Again, if you have any questions, use the QA system through Google Hangouts. Otherwise, you can email me if you have any questions. I'm watching my phone right now. Okay, so then we have this red portion right over here, this ampicillin. This is an ampicillin resistance gene, which means if this plasmid is inside a bacteria, it's going to make it resistant to ampicillin. And what is ampicillin? It's an antibiotic. It's what's used to kill bacteria. When you get sick, you take erythromycin, kanamycin, Z-Pak. You can even take penicillin and ampicillin to kill bacteria. This gene makes it resistant to ampicillin, and what it's actually doing is this gene produces an enzyme which breaks down ampicillin. So when it produces this enzyme, it kind of spills it all around the bacteria, and it will break it down, so then the bacteria is not exposed to ampicillin. So in Lab 7, what you're looking at, the GFP plates, basically what they did was they took two plasmids. You had one plasmid that had the green fluorescent protein, and then you had another plasmid that had a promoter site for the green fluorescent protein, and it also had an ampicillin resistance gene. But what you really don't know, what you may not understand, and I'm not sure if the other TA went over this, is how did that gene get put into another gene and then get put into the bacteria? And so I'm going to kind of break it down step by step the best I can. Please ask any questions if you get lost, or wait until next week when I will be in class and we can kind of go over it again. So basically, kind of out of order here. Here we have a piece of a sequence, GAATTC-CTTAAG. What this is a sequence that's inside the gene. In fact, this is a restriction site. What that means is there are enzymes that come from bacteria that can cut up DNA at very specific sites. In this case, the enzyme called EcoR1, and yes, it did come from E. coli. It's able to recognize this very specific sequence, GAATTC. And when it recognizes it, it cuts it. And it cuts it in the way that you see the dotted lines. And what it does is it forms what they call sticky ends. This is really important. I'm not going to go into too much detail about the sticky ends, but there's different types of cuttings. You have a blunt end and you have sticky ends. Sticky ends are the easiest way to cut DNA and to make DNA come back together because you have that overhang. Those T's over here are looking around for A's to bind to. So if you mix two pieces of DNA together, they're most likely to find each other. Okay, so let's now going back over here. So let's just say we have our tube of plasmid, bacteria DNA that we want to cut out a gene, or just cut it in half. And we have another gene that we want to take, so let's say GFP, as you saw in the previous lab, and you want to put it into another plasmid. That is what they call recombinant DNA. So essentially, we have two pieces of DNA. We've cut out the gene of GFP, and we can now put that into another plasmid, say the plasmid with ampicillin resistance. Don't need to go through all that. So, in order to make these two genes then come together, we need another enzyme called DNA ligase. I think you guys have gone over that in class. Ligase creates those phosphate backbone bonds. At this point, we just have the A's binding with the T's, and those are just through hydrogen bondings. But we need ligase in order to complete the phosphate backbone. So this is more of a real-life scenario, especially what you saw in Lab 7. PCAN-R plasmid. It has a kanamycin resistance gene in it, and it has a RFP, you guys were using GFP. It's still a fluorescent protein that's just different colors. If you guys go online and look up San Diego Sing Lab, you can see all the colors that they have come up with, and they were the originators of creating GFP. Then we have this P-era plasmid. It's got an ampicillin resistance gene. It's got an ERA C, a PBAT promoter, and you see these two labels, BAMH1, HIN3, and we have the same one, HIN3 here and BAMH1. Those are restriction sites, very specific sequences that enzymes, very specific restriction enzymes, will find. And so if you mix in with those plasmids these two restriction enzymes, you can cut the DNA. Just like that. Now essentially what you have is 4 pieces of DNA. Going back over here, after we've cut those 2 pieces of DNA, we now have 4 total. We have 1 all the way on over here, and we have this little piece of RFP, and we have this PR, all this over here, and we only cut just a really small area. That may only be 20 or 30 base pairs. Nope, 40 base pairs, close enough. So we're dealing with really small volumes. How do we know that we've actually cut our DNA? We do an assay called DNA electrophoresis. And essentially what that is doing is we're taking a gel, agarose, remember agarose, those horrible math computations that I had you do in Lab 5? Agarose gel. That is what we use to do a DNA analysis. If you guys have ever seen CSI and they have those pictures of different bars and they're talking about genetics and you see the different lines on this graph, that is a gel electrophoresis, just like what I have here. So essentially what they're doing in this little experiment right here, we have M for marker, or also known as the ladder. Ladder may be a little bit easier to understand. K plus, K minus, A plus, A minus. So if we go back, our PKR, that's our K, and PRA, that is our A. And what we do is we always want to make sure we're actually cutting our DNA. So we always have a control. That's why there is a P plus and a P minus. P plus means that is the part of the gene that you've actually cut. The K minus, that is the control. You don't add any restriction enzyme. The P-era, which is the A plus, that is where you actually add in the restriction enzyme. And the A minus, that is our control. We have not added any restriction enzyme at all. Okay? So now what you're going to do is you're going to run electricity through this little gel. And with it, we want to make sure that we have this marker, or this ladder. It's our known control. So when we run electricity through it, we're going to see DNA of these different K plus, K minus, A plus, A minus. And now using our control, our marker, we can figure out exactly what sizes of each band we see here. And this is exactly what they do on CSI, all those movies, on TV shows. So if you look at the K plus, okay, we see a band right over here, and it's between 1,500. What do these numbers mean? These numbers are the base pairs in DNA. So this band right here is telling us that we have a size of DNA in between 500 and 1,000. I would roughly approximate maybe about 750, give or take. And then we get a band all the way up here. And I would roughly say it's about 8,000 base pairs. Now that's our cut DNA. Our uncut is far bigger. And essentially it should be our 2 cut pieces added together, the base pair size. So if we have 8,000 plus 750, we should come up with about close to 9,000. I'd say this might be a little bit higher than 8,000, give or take. And we go over here to our A plus. This is about 7,000, give or take. And then we got this little 40 base pair over here. And now this picture over here, this is what an actual gel, what it looks like when you run electrophoresis. And this is what we're going to be doing next week. So I'm going to go into far more gruesome detail next week about this. Okay? So, a little bit more graphical interface to show you what's happening when we're taking our two pieces of DNA and we're putting them together. So we have this RFP gene right here, and we have our P-ERA gene down over here. These enzymes, BAMH1 and BAMH1, HIN3, HIN3, they've created sticky ends. And remember I was saying sticky ends make DNA coming back together far easier? This CTAG is looking for GATC. And look at that over here. TCGA is looking for AGCT. And they just combine together. Now at this point, they're still only being loosely held by hydrogen bondings. They can easily break. You need an enzyme called ligase to come and finish off those phosphate backbones, so you can see those bonding together like that. And now we have created a plasmid called P-Glow that will fluoresce under UV light. And it also has a PBAD, beta-lactamase, origin replication, ERA-C. So now this is the really important part. This is what's going to be on the quiz. Okay, this is similar to your pre-lab that you had. You were only given 4 different plates, 4 different conditions. Essentially, we have our P plus plates and P minus plates. P plus stands for our DNA fragments or plasmids that have been transformed. In other words, we have successfully taken our GFP gene, or in this case RFP, we have put it into the P-ERA gene, so it has an ampicillin resistance to it. And then we have successfully taken that DNA and put it into the bacteria, just like last week's natural transformation. Okay? So you guys are going to predict what you're going to see, what type of growth, and where. The other TA should have gone over different types of growth. A lawn versus some growth compared to no growth. So, think about this. A P plus that has been successfully transformed, and our P minus, that means this is bacteria that has not been transformed. It does not have the gene for GFP or ampicillin resistance. This is really critical that you understand this, so please ask me any questions that you may have. This will be on your quiz. Okay? So, think about this. LB. LB is just plain food. It's food for bacteria to the E. coli just to eat and grow and live. If you have successfully transformed bacteria to have an AMP-resistant gene and a GFP gene, will it grow on the LB plate, and what type of growth? I'm hoping you guys are thinking you should see a lot of growth. There is no selection. There's nothing that's forcing the bacteria to die off. LB is just food, so you'll see a lawn. And you will see no GFP because you need something else to make it glow green. And I'm going to go into a little bit more detail about that. Actually, I should probably go into more detail about that now. So, let's go into that right now, and then we'll go back. We have our RFP gene in our P-ERA plasmid. Right now, with the RFP gene inside this plasmid, you're not creating any RFP, or in your case last week, GFP. Why? Because there is another gene called ERA-C that is being produced. It is producing a gene that is actually going to inhibit the production of RFP. This ERA-C protein creates a hairpin in the DNA, so it binds to both sides of the DNA. And when it's binding to both sides, it doesn't allow room for RNA polymerase to bind to this RFP gene to make mRNA, to make protein. So it inhibits it. But if you have a plate that has a rhabinose, or in your case for your lab, it was IPTG, which is also a sugar. It's just a different type of rhabinose, different type of sugar. Rhabinose is also sugar. As long as you have a rhabinose, it's going to bind with this Ara-C protein. And when it binds with the Ara-C protein, it changes the conformation, it changes the shape. And when it changes the shape, it binds with RNA polymerase, and then it kind of helps to sit right onto the PBAT promoter, which then allows RFP gene to be created. And it creates RFP. This is GFP. This is what you've actually looked at in the lab. So now let's go back real quick. Right back here. You already saw the answers, but you're going to be quizzed on this, so I want to make sure you understand it. So we have 3 different types of plates here. LB, LB ampicillin, LB ampicillin arabinose. Okay, hope everyone understands that. This LB plate just has food on it. LB ampicillin has food with the antibiotic. And this plate has LB, the food, with the antibiotic, ampicillin, and it also has that sugar, arabinose. So, what kind of growth do we expect to see on LB? We already went over that. We're supposed to see a lawn. Nothing discriminating. Everything should live on it. Now, we have this LB ampicillin. This has antibiotic on it. Should all of this bacteria grow on it? Well, think about this. When you, last week, or 2 weeks ago now, did you see lawns on all your plates? No, you didn't, because not every single bacteria cell picked up DNA. It's all by random chance that it's going to pick up the right piece of DNA. So you're only going to see some growth. Only the bacteria that got that gene will grow. Are you going to see any red on here? No, because there is no arabinose to make the red gene. Make sure you understand that. There is no red or green fluorescence because there is no arabinose to turn that gene on. You can think of arabinose as a light switch. On this plate, again, you see a little bit of growth, and you'll see some red fluorescent protein, or some green in your case, as you saw in lab. And this is also because when you do your ligation, not every single piece of P-Ara will get the RFP gene, or the GFP gene in your case. So you have 2 things kind of happening by random chance. We're hoping that some green fluorescent protein or red fluorescent protein will get into the P-Ara, and then we're hoping that that P-Ara will get into the bacteria. For the plasmids that did not get the red fluorescent protein, they still have the ampicillin-resistant gene. That's why we see a mixture of red and white. Now we go over down here to our control, our P- plates. Again, we expect to see a lawn on LB because it's just food, but you should see absolutely no growth on the LB amp plates. Why? Because it does not have any plasmid that has the ampicillin-resistant gene. So I want you to study this very carefully. This will be on your quiz. Alright, so we've gone over that, we've gone over that, blah, blah, and green fluorescent protein, which is what you guys saw. We already went through this, and that's it. So, I hope you guys understand. Again, I apologize for not being in class on Wednesday. If you guys have any questions, please feel free to email me or call me, email me any questions or ask me in class, and we will go over it then. This is really important. Again, I apologize for not being there. This next coming week, we're going to do a PCR. So I don't know if the TA went over that, but we're going to go into more detail. The lab kind of takes a few steps further, which is a little bit confusing. I don't care about that as long as you guys understand basically what a PCR is. So If there are no other questions, I will end this broadcast right now. I will still stay online in case you guys have any questions for another 30 minutes. But this video will automatically get put up on YouTube, and I will put that video, the link, on the forum, so you guys should get the email right away. Any questions? Nope? Okay. Well then, thank you for watching the video, and I will see you guys later this week. Have a great day. Bye.